Good afternoon, everyone. Okay. So this morning you've heard about the amazing capabilities of the ultra-high gradient strength MRI system enabled for diffusion MRI and beyond. And what I'm going to show you next is an ex vivo data resource we've collected on macaque and human brain enabled by these systems, including the Connectome 2.0 here. Okay. So we collect this data as part of the large-scale imaging of neural circuits project. In previously Anastasia's talk, you've heard about this project. The LINK project involves diffusion MRI data collection and also multiple optical and X-ray microscopies on the same specimen. And the overall goal of the project is to develop and validate, develop and cross-validate imaging techniques for imaging the axonal projections in the human brain, and use these techniques to develop a system of the fiber pathways in order to improve the targeting for deep brain simulation. And this rich data resource, we hope, will also inform biophysical modeling with diffusion MRI, which is the only non-invasive methods that can be applied in vivo. So as the first step of the whole imaging pipeline, our goal here with diffusion MRI is to comprehensively sample the brain using those state-of-the-art ultra-high gradient systems, including the Connectome 2.0 for imaging the larger human brain. And our goal here is to develop protocols that is optimized for both fiber tractography and also tissue microstructure mapping. And so you've heard in the morning talk, ultra-high gradient strength is really critical for shorten the echo time and also enable higher B-value imaging. And all these are even more critical for ex vivo imaging because due to fixation, the fixed tissue has a substantially shorter echo time. So here, shorter T2. So here, a shorter echo time is even more critical. And the fixed tissue in the room temperature also has a much lower water diffusivity. So this requires us to use even higher B values in order to achieve the same contrast in vivo. And last but not least, in fixed tissue, a part of the water the immobile water become non-negligible in fixed tissue, and this is contribution to the diffusion MRI signal must be considered in order to get more accurate tissue parameter estimation, and this requires us to use even higher than standard high B value imaging. So that's why ultra high gradient here is really the key. And in our prior work on the preclinical system for macaque imaging, we used the 3D EPI, and we also find that using a high number of segments can be very effective in terms of reducing the echo time and improve the image quality overall. And using multi-segments means we will have longer acquisition time. So here, temperature control is really critical for the imaging the larger human brain, and we used a custom-built ex vivo coil developed here, and so it has a ventilation system during the long scan hours for temperature control. And so just to quickly summarize with what we have achieved, we developed high resolution protocols primarily for orientation mapping and the fiber tractography, and achieving 400 micron isotropic in the human brain and 250 micron isotropic in the macaque brain, and both with ultra high B values. And additionally, we have lower spatial resolution protocols that is sampled across different diffusion times and echo times. So it can give us a richer data set for more detailed microstructure modeling. And so we are sharing this data resource on the Dandy archive. And if you're interested, you can also look at our preprint. So what I'm going to show next is a few analyses we have performed to demonstrate the quality of the data. And so for tractography, Chiara later will show more detailed analysis. And here, I'll just give you an example of the tractography in the hippocampus. This small anatomical complex structure deep inside the brain. And with our high resolution data, we are able to track the major the major pathways projected from the entorhinal cortex to the subiculum and to the dentate gyrus, as well as the smaller mossy fibers connecting the dentate gyrus and the CA3, and also the sharper collaterals from CA3 to CA1. In both human and a macaque. So this really demonstrates that our data supports detailed anatomical investigation and also cross-species comparisons of structure connectivity. On the human side, we have effective higher resolution, and we also demonstrate that this data can help delineate the cytoarchitectonic layers. And for this demonstration, we also use the soma neural density imaging model. And here we use the probabilistic estimator with priors optimized for ex vivo imaging using the microstructure Julia toolbox. So we estimate the intrasoma signal fraction and the intraneural signal fraction as indices of the cell body and the neural density measures. And if we zoom in onto the hippocampus, you can really appreciate how well the soma density maps can help delineate the different substructures and subfields of the hippocampus. An example I'm showing here is the granular layer of the dentate gyrus. And from Niso staining, we know this is a cell dense layer here. So this really demonstrates the high resolution and also high SNR of the data. We also looked at the cortex, and to help visualize the microstructure variations in the cortex, we applied a layer segmentation methods that is previously developed on even higher resolution structure scans from the 70. So we applied these methods to our mean average the B0 images to segment the supragranular and the infragranular layers. So the microstructure variations within the cortex also closely resemble the Niso staining summarized in this book chapter. And so, for example, you can see the lack of cell dense layer 4 in the motor cortex versus the presence of the cell dense layer 4 in the visual cortex by looking at the cell body fraction near the boundaries of the supra and the infragranular layers. So now let's move on to the multidimensional data. And for this demonstration, we compared a single echo time SANDI estimates versus multi-echo time SANDI estimates. So this model was first demonstrated a few years ago on Connectome 1 data in vivo in collaboration with Dr. Marco Colombo at Cardiff University. So here, what we see here is the multi-echo data really is able to detect the larger cell bodies in the motor cortex and also the smaller cell bodies in the cerebellum. And it is pretty consistent across the four macaque brains we've imaged so far. On the human white matter microstructure imaging, we also show that the axon diameter index estimated from the 2 diffusion data can capture better the variabilities across the white matter in comparison to estimates from a single echo time. So just to summarize, this data resource offers a rare combination of high resolution and also ultra-high diffusion weighting, which is really enabled by those ultra-high gradient systems. And additionally, it provides lower resolution but multidimensional data for more detailed tissue microstructure mapping. So it has a lot of application potentials. Like we've demonstrated, it can be used to do cross-species neuroanatomical investigation, and also with the multiple resolution, multiple dimension data, it can also be used for methods development, such as super resolution modeling. And also, we can build template or atlas using this data set, which is can be helpful for clinical studies. And last but not least, with the data coming from all the other modalities, this will become a foundational resource for also validation and improvement of non-invasive diffusion MRI. So with that, I would like to thank the funding resource BrainConnects project, and also the additional fundings, and also all the collaborators and the contributors who has made this work possible. Thank you.
Great, thanks so much, Ting. And then we have a companion presentation that will be given by Chiara Maffei, who's also an instructor in radiology here at the Martinos Center on ex vivo ultra high resolution diffusion and tractography, high definition subcortical pathways at 400 microns. Thank you, Chiara.
Thank you, Susie, for the introduction and for the invitation to speak today. My goal is to show you what we can do with tractography when we leverage these very high resolution diffusion MRI data in humans that we acquired as part of the LINK project and that Ting just showed to you. So the basal ganglia thalamocortical circuitry is a large fiber system in the brain that supports critical function across motor and cognitive domains. And it's composed of three main pathways: the hyperdirect, direct and indirect pathway that carry information from the cortex through the basal ganglia, and then back to the cortex. These cortical subcortical connections are somewhat easier to reconstruct using tractography, but these subcortical components here are too small to be resolved using conventional diffusion MRI. What makes them particularly challenging is not just their size, but it's also this very complex subcortical region where multiple fiber pathways merge and overlap, and also have to cross these larger fiber systems here, like the internal capsule. So despite their importance across motor and psychiatric diseases and their involvement in neuromodulation application, there is at present no tractography resource to visualize them. And clinical and research application have to resort to either classical histological atlases or synthetic data. So using the very high-res data that we acquire in the Connectome, we could reconstruct all these components for a total of 44 pathways, 19 of which subcortical and 25 subcortical-cortical, so that we could generate the first tractography-based comprehensive atlas of the basal ganglia circuitry. This animation here shows the classical flow of information from the cortex through the basal ganglia and then back to the cortex, overlaid on our tractography reconstructions. So we know from invasive tracer studies in non-human primates that these connections are topographically organized. So to assess the anatomical validity of our reconstructions, we compare the organization of the connection against this known gradient from the literature. We first clustered the subcortical-cortical components based on their cortical termination in the frontal cortex, and confirmed that the organization of streamlines within the internal capsule, the structure here, replicated the same organization that we see in tracer studies, with ventral and orbital connections always coursing inferior to like more dorsal and motor and premotor connection here. We also confirmed that just caudal to the anterior commissure, which is this dark red structure that you see here, this streamline is split into two components, one that enters the thalamus and one that continues down the brainstem. And you can appreciate that here in this short animation where you can see this entering the thalamus and then continuing down the brainstem. We then mapped the termination of this connection onto their subcortical components, so the putamen, STN, and thalamus, and could replicate the same connectivity gradients with this rostral to caudal, but also ventromedial to dorsolateral organization that we know from the literature. Now, focusing particularly on the subcortical components of this atlas, we could accurately reconstruct not only the classical basal ganglia connections, including some very small projection from the globus pallidus that had so far only been described in tracer studies, but we could also reconstruct a series of adjacent limbic pathways that are also relevant for various motor and psychiatric diseases, and that are usually not included in tractography reconstructions because they're too small. So then finally, we tested the clinical utility of our ex vivo dataset to work as a normative data in vivo deep brain stimulation studies. So we mapped our atlas onto the MNI space. We partnered with neurosurgeons to select stimulation hotspots from published DBS studies. And we generated hotspot-specific networks and derived connectivity fingerprints by looking into the degree of engagement of each tract within the atlas relative to each hotspot. Overall, we observed that hotspots that are correlated with improvement in motor symptoms show the largest engagement across motor pathways, in blue here, whereas like non-motor hotspot show larger engagement of limbic and association tracts, which is what we would expect. So the consistency of this pathway symptom relationships confirms the validity of the atlas and also demonstrate its potential to identify specific white matter connections that could mediate DBS efficacy. So just briefly to conclude, we show the feasibility to reconstruct very small subcortical pathways with unprecedented accuracy using this very high-res data. We proved the anatomical validity of this atlas in replicating organizational rules that we know from invasive tracer studies in non-human primates. And we also showcased the potential of this dataset to work as a normative dataset in vivo DBS studies. If you would like to know more about the study, we do have a preprint out. You can scan the QR code there, and you can also access the data that we made publicly available on Dandy, or you can interact with the atlas on our link gallery website that you see here. I'd like to thank all the people that made this possible, you for your attention, and I'm happy to take any questions.